Hello students, welcome back to a new chapter. The chapter is sexual reproduction in flowering plants. You know that out of the angiosperms and gymnosperm we have the angiosperm known to be the flowering plants. Yes or no? Now the students, flower has been intimately related to the humankind or the mankind since time immemorial. Flower has been used to express sorrow, to express joy, to express love and so many other areas. Uh, this diagram represents the structure of a bisexual flower. I hope you all know this very simple diagram. We have dealt with this labelings and the structures in class 11. I'll be skipping this section very fast. Now the main thing is how the flower comes to know that a plant comes to know that it will flower. Now, there are several hormones. There are several structural changes. Now one such hormone is a florigen. A hormone called florigen, florigen. This hormone is present in the leaves. Very interestingly, the hormone is present in the leaves, and this hormones actually helps or actually initiates the differentiation of the birds. Buds. The birds are present in the various parts of the plant. It helps in the differentiation of the birds into flower. And what happens, initially a floral primordium, a floral bud is formed, and that floral bud gives rise to a flower. And as you all know that a typical flower has a typical bisexual flower in fact has 4 floral whorls. From outside to inside, they are the calyx, corolla, androecium, and gynoecium. Of these 4, the calyx and corolla, they are considered to be the non-essential parts of a flower. Non-essential, since they're not directly involved in reproduction. However, they have some role. But the androecium, the male part, and the gynoecium, the female part, these two are responsible for reproduction. These two parts contains the reproductive organ, hence they are called the essential parts of a flower. Today we'll see very slowly the different structures related to the stamen, microsporangium, and pollen grain. The stamen is a male reproductive part, as you can see in the diagram. Yes and no? This is a stamen. This is a stamen. A stamen consists of a filament, a long filament. This is a long filament, a long filament here, a long filament. Okay, and this filament has at the anterior portion a structure which is called anther. Yes or no? This is an anther, and this anther is bilobed in structure. Bilobed, L-O-B-E. Lobular structure. So this is a bilobed structure having 2 lobes. And this filament at the end is attached to the it may be attached to the petals, it may be attached to the thalamus, it may be attached to the tepals. Okay, so there are different kinds of attachment of the filament to the flower. Now if I make a section of this anther, you can see one diagram here. If I make a transverse section of this anther, you can find that in one of the lobes or each of the lobes there are 2 structures, 2 caps. Yes or no? 2 thecus. Each lobe has 2 thecus, hence called dithecus. In each of the lobe there are 2 thecus, 1, 2. Here another lobe, 1, 2. Each of the lobe has 2 thecus called dithecus. And since a typical anther has 4 theca or 4 microsporangia. They are called the microsporangia because the microspores or the pollens are formed within the structure. So 4 microsporangia, hence it's called tetrasporangiate. I hope the name is not mentioned here. Is it mentioned? It is called tetrasporangiate. So let me repeat. An anther is bilobed in structure. In each of the lobe there are 2 microsporangia. So together 4 microsporangia, hence it's called tetragonal. This 4-sided tetragonal structure, they consist of 4 microsporangia as it is highlighted in yellow. So it is called tetrasporangiate. Yes and no? And within this microsporangia is a plural word. Microsporangium is a singular word. Within this microsporangia, the pollen grains or the microspores are formed. Now let us see in detail the structure of one of this microsporangium. To know the structure, I will directly scroll to the diagram first and then I will explain. Look at this diagram, students. Okay. So this is one transverse section, the figure number A. This is one transverse section of an anther. You can see 1, 2, 3, and 4 microsporangia. We have 4 microsporangia. This diagram is very important, students. It has been often asked in exams in CBSE as well as school level examinations. Now, if I zoom into these walls, these layers of the microsporangium or microsporangia, what you can see? You can see 4 walls. Very important. The names. The outer is called the epidermis. This is wall number 1. The outer wall is called the epidermis. Wall number 1 is called the epidermis. Then we have the endothecium. Next to epidermis is the endothecium from outside to inside, and next to the middle layers, which can be 2 to 3 numbers, middle layers, so it's a S plural, middle layers 2 to 3 number, and within the middle layers we have a layer called teptum. As you can see, this red color, this nucleus teptum are the rich in cytoplasm, food laden cells. So we have the 4 layers. The outer layer is the epidermis. Next to epidermis is the endothecium. Next we have 2 to 3 middle layers, and then we have the teptum. And interior to the teptum, now look at this diagram A. Interior to the teptum we have this porogenous tissue, homogeneous tissue known as porogenous tissue. These tissues, they undergo meiosis. And ultimately give rise to the microspores, also called pollen grains. Remember, students, very important. So what are the 3 layers? Epidermis, endothecium, middle layer, teptum. Now, if I come to the function of these layers, let me scroll up. Here I have highlighted very important section. If I come to the structure, the function of these layers, the outer three layers perform the function of protection and help in the dehiscence of the anther to release the pollen grains. I have highlighted this line, students. They help in the protection of the anther and also in the dehiscence. Dehiscence means the rupture, the bursting of the anther to release the pollen grains. And the innermost layer, which is called the teptum, or innermost wall, here it's called the teptum. It nourishes the developing pollen grains. Okay, students. So if we come at the definition of microsporogenesis, it's a very simple definition whereby you find the word genesis means formation, microspore means the pollen grains. So the cells of this porogenous tissue, which are present in the center of each of this sporangium, the cells of this porogenous tissue, they undergo meiotic division to form microspore tetrad, and the process is microsporogenesis. Microsporogenesis means process of formation of the microspores. Microspores are nothing but the pollen grains, and pollen grains are formed from the sporogenous tissue that occupy the center of the anther. The center of the microspore is the center of the microsporangium. This region, this center. Okay. This is the actual diagram. So you can see the anther has dehisced. So bursting, these pollen grains are coming out. The pollen grains are released like this, okay, by the bursting of the anther, which is called dehiscence of the anther. And dehisced anther. Okay. Here you got the definition of pollen grains. Now, students, very interesting to note when the pollen grains, the pollen grains are formed, or the microscopes. In fact, now they are called microspores, not pollen grains. When the microspores are formed, they are formed in a cluster of 4. 1, 2, 3, 4. Look at this diagram. A. They are formed in a cluster of 4 microspore. This is called microspore tetrad. The word tetra means 4. So when the microspores are formed, each of these sporogenous cell, remember, each of the sporogenous cell will undergo meiosis, and you know that in meiosis one cell gives rise to 4 daughter cells. So from one of these sporogenous cells we got these 4 cells, which is called microspore tetrad. Okay. In this diagram, you can see the different pollen grains collected from different flowers. The pollen grains have spiny outer ball. We'll come to this structure also one by one. Now, if you see the structure of a pollen grain, generally a pollen grain is of 25 to 50 micrometers in diameter, very small in size. The outer layer is called the exine. Very important term, students. This section, this highlighted section is the most important part of this chapter. The outer layer is called the exine. Exine is made of sporopollenin. It is made of sporopollenin, which is one of the most resistant material known, organic material known, and it can withstand high temperature, high strong acids, strong alkalis, and no enzymes that can degrade sporopollenin is so far known to us. And this is for this reason. This is for this sporopollenin that pollen grains are preserved as fossils. Listen, students, very interesting. Now the exine, which is the outer layer, the exine is not continuous. The outer layer, which is exine, is not continuous. It has some gaps. Though it is not understandable from this diagram, I'll be very soon sharing another video where I will show you the exant, intent, the gaps. Okay, the gaps. The gaps are called the jump pores, where the sporopollen is absent, and through this jump pores, through this jump pores, the pollen tube comes out, or the pollen germination takes place. The next to the exant is the intine, which is the inner layer. Intine. These are the two walls, or the two layers of a pollen grain. Exant, the outer wall, and the intine, the inner wall. Okay. Intine is, however, it is thin, continuous, made of cellulose and pectin, also called pectocellulose. Now, within a pollen grain, what structures are present? This I'm skipping, students. I'll be very soon sharing another video where I'll be dealing with the structure of a pollen grain and the mechanism or the process of formation of a pollen grain. I will come to this section. Now, pollen grains are also known to be allergic, which is called pollen allergy. Okay, sometimes you sneeze, you don't know the matter of sneezing, okay, because you have inhaled pollen grains. Pollen grains are known to be allergic. Certain pollen grains are known to be allergic in some people. Okay. Now, pollen grains also have nutritive value also. Okay. Pollen grains, you know that the honeybees, they look for honeys, not only for honeys but also for pollen grains, because the worker bees, they are fed pollen bread. That is a mixture of pollen grains and honey. Okay. So these pollen grains, or the pollens, they are made in form of tablets, they are made in form of syrups, and they are used as nutrient supplement. Okay. They are even given to the race horses to improve their capacity, to improve their agility. Last segment of this topic is, once the pollen grains are shed, once the pollen grains are burst out from the anther, they have to land on the correct flower. Yes or no? Means a pollen from mango must fall on the stigma of a mango. Yes or no? The same species, then only it can germinate. So, but what is the time? Every pollen grain, every species has got its, the time. For example, here it is said that in case of wheat and rice, the viability is only for 30 minutes. 30 minutes of the release, means after 30 minutes, the viability is lost. If the lifespan of the pollen grains is lost after 30 minutes, that means within 30 minutes the pollens of rice and wheat must come in contact with the stigma. Okay, but in case of some other members of Rosaceae, Leguminaceae, Solanaceae, these are the different families of the plants. They are viable for months. For months they can remain active. Okay, so you have heard about storing of semen, that is called the for artificial insemination. Semen can be stored in liquid nitrogen. Similarly, pollen grains can also be stored in liquid nitrogen at -196 degrees Celsius, and that is called pollen banks. So pollen banks, when these pollen grains can be used for the pollen glands that helps in the crop breeding programs by the farmers to produce improved variety or hybrid variety of crops. That's all for today's video, student. I will be very soon sharing you the process of the formation of a pollen grain, a mature pollen grain from a microscope. Thank you. Have a nice day.